Greetings, scientists, and welcome to this supplementary lecture, online lecture for what we would have covered in class today being Thursday. So we're continuing this conversation regarding internal transport. So last time we focused on water relations, but today we're going to focus on gas exchange. We will talk about plants and animals and a few other organisms, including some bacteria and fungi, but the focus will be on plants and animals. This is the list of objectives that we'll be covering. So this is going to be a pretty dense lecture today, but I'll try to walk you through all of it. And of course, if you have any questions at all, please feel free to email me or talk to me in class. Oh, I should probably point out that I was planning on including this material on the exam coming up on Tuesday, but I've decided to not include this information. It makes me uncomfortable to not be able to cover this in class and answer any questions. So we'll put this on to the exam 2's material. So everything from the beginning of the semester up to Tuesday's lecture, the previous lecture, is what will be on the material. So we've been talking about internal transport. So what all is being transported throughout plants and animals and their bodies, as they were. So clearly, many gases are being transported. So oxygen and carbon dioxide, water, which is what we covered last time, and then all kinds of nutrients and waste products. So today we're going to focus on gas exchange and what's happening with those systems. So in class, we would have had a team discussion on why do you need oxygen, right? So most people respond with, well, you need it to respire. But we're trying to get you to think about it on a deeper level, right? So what exactly is happening in your body or in the bodies of plants and animals and microbes? What do you do with oxygen? So the team discussion would have been, so cyanide poisoning inhibits the action of the electron transport chain in cellular respiration, and has the same result as lack of oxygen, also known as histotoxic hypoxemia. So if you don't have oxygen, what actually causes your death? So think about that for a minute, maybe hit the pause button, do yourself a favor and test yourself, hit pause, think about that answer, and let's see if we end up in the same place. Okay, assuming that you actually did that. So what's the importance of oxygen? Well, as you may remember from 111 or subsequent classes, maybe in cell. So oxygen is supremely important in the cellular respiration cycle, right? So it's the last step in the electron transport chain. If you fail to have oxygen, or if you have a low level of oxygen, that's going to inhibit cellular respiration. And ultimately what will happen is a shutdown of all of your tissues. So your tissues, your cells would die, your tissues would die. And so depending on exactly the nature of the lack of oxygen, you're going to have widespread organ shutdown. And then this precise explanation for your death might vary depending on which organs shut down first. But ultimately, all your organs are going to shut down, and it can be fatal. So now let's describe the factors that affect oxygen and carbon dioxide levels in the atmosphere and in the water if you're talking about aquatic organisms. And so remember, at the beginning of the semester, we talked about basic principles involving diffusion and facilitated diffusion versus active pumps. And so you'll recall that some gases can diffuse through membranes without costing any energy, without costing ATP. So let's dive into what happens with gas exchange. So if we were in class, we would have had some iClicker quizzes. So water with high levels of decomposition will have blank levels of dissolved oxygen. So if you have high levels of decomposition, will you have higher levels of dissolved oxygen, unchanged levels of dissolved oxygen, or lower levels? Think to yourself the answer. So with high levels of decomposition, you'll have lower levels of dissolved oxygen. So basically you're in an anoxic situation, so not enough oxygen to help you. What about this? If other factors are equal, which of the following environments would have the highest level of dissolved oxygen? Test yourself first, look through these answers. What do you think? So the answer should be D. Cool freshwater is going to have the highest level of dissolved oxygen. Okay. So gas exchange, in order for gas exchange to occur, you need to have an environment where the gases can diffuse across membranes. So gas exchange between the environment and cells is based on diffusion, and in both air and water, oxygen and carbon dioxide diffuse from regions of high partial pressure to regions of low partial pressure. So this is in direct alignment with what we were talking about at the beginning of the semester. And so what this figure is showing you is sort of exemplifies that. So if you're talking about human respiration, right? So oxygen needs to diffuse into the circulatory system through the lungs. So it's gonna cross the membrane of the alveoli and go into the circulatory system, so into the capillaries. And at the same time, CO2, so carbon dioxide, is going to diffuse out. And generally speaking, oxygen levels are higher in the atmosphere and lower in tissue, so they're often going to, they'll actively, or they'll not actively, but they'll diffuse across into the tissues. Carbon dioxide is often higher in tissues than it is in the atmosphere, and so it will diffuse out. And this is going to follow partial pressure gradients. Oh, sorry, I got ahead of myself. So here I say the same thing. So oxygen is higher in the environment, low in tissues. Carbon dioxide, high in tissues, low in the environment. So you can actually calculate the rate of diffusion following Fick's law of diffusion. So this formula that you see here. So let me just walk you through this. So the rate of diffusion depends upon several different variables. So what we're representing here as K is simply a constant. Now that constant is going to depend upon the solubility of the gas that in question and the temperature at which this reaction is happening. But for now, just consider it a constant. This A represents the area for gas exchange. So you might have a smaller area or a larger area for gas exchange. P2 minus P1, so this is the difference in partial pressure of the gas on either side of the barrier. So I usually think of this as P2, where's my cursor? There you go. P2 is usually the partial pressure outside, so in the environment, and P1 is on the inside of the respiratory membrane. So you can think of it as being, P2 is outside the lungs, P1 is inside the lungs. And D here is the thickness of the barrier to diffusion. So if you have a really thin membrane that you're going through, or a much bigger membrane, or a series, or maybe several different layers of cells that you have to go through, that's going to impact the rate of diffusion of gases into and out of the body. So let's think about what happens if you try to adjust each of those different parameters, right? So we're looking at them in sequence. So if you increase the diffusion constant, the partial pressure inside or outside the tissues, the area for gas exchange, and then the distance. And if I go backwards and look at this formula again, so think of it as being simple algebra. So what happens if you increase each of these different variables? What's going to happen? So if you increase K, what's going to happen? If you increase K, you're going to increase the rate of diffusion all the way around. So if you want to think about this simply, just imagine all the rest of this as being a single number. Doesn't matter what number it is. If you increase K and multiply together, clearly that's going to increase the value of the rate of diffusion. The same will be true if you increase the area of gas exchange. Bigger area means higher rate of diffusion into that body. Now here's where it gets kind of interesting. If you increase P on the outside, right? If you increase the partial pressure of gas on the outside, that's actually going to end up increasing the rate of diffusion all the way around. Okay, and think of it like this. So let's say P1 is, doesn't matter what number it is, just pick a number. It could be 5. And then let's say D is 10. Okay, so if P2 is 10, 10 minus 5 is 5, and then 5 divided by 10, right? Do your simple math. Now, think about if you increase this number. All that's going to do is increase the entire value of the equation when you multiply it across. Simple algebra, so it's going to increase the rate of diffusion. However, if you increase the rate of gas on the inside of the body, that's actually going to result in a decrease of the rate of diffusion into the body. Okay? Because you've increased the pressure on the inside, okay? That's going to slow, remember, partial pressure, so gases are naturally going to diffuse across from areas of higher pressure to lower pressure. If you increase the pressure on the inside of the body, that's going to slow the rate of gases going into the body. Similarly, if you increase D, the thickness of the barrier, that's also going to slow down the rate of diffusion into that body from the outside environment. So most of these variables, if you increase them, they will increase the rate of diffusion. The two that will result in a decrease is if you increase gas, partial pressure of gas on the inside of the body, or if you increase the thickness of the barrier. Hopefully that makes sense. If you have any questions, I'm happy to answer them. Okay, so based on that part of the lecture so far, so based on Fick's law of diffusion, why is breathing more difficult at high altitudes, such as at the top of Mount Everest? So pause this, maybe test yourself, think about what the answer would be, and then click back on when you're ready. So if you've come back, so based on this law of diffusion, right? Now we haven't actually talked about altitude just yet, but you'll know from having done the reading that partial pressures are lower, right, at high elevation than they are at low elevation. If the partial pressure is lower, then what that means is that the rate of diffusion is going to be lower as well. So it's going to be harder to get oxygen into your body, and it's going to make you feel out of breath. Actually, if it's extreme, it'll have more extreme symptoms, but it's often difficult for people to breathe, or they find that they run out of energy quickly, say if you're skiing at high elevation. And that's because the partial pressure is lower. So let's look at that in a little bit more detail. So if you're trying to calculate partial pressures, this is simply the fractional composition multiplied by the atmospheric pressure. So if you look at this graph of Mount Everest, you can see that here's atmospheric pressure right here. So pressure is always higher at lower elevations. So it's 760 millimeters mercury, compared to at the top, it would be perhaps 250. The formula is easy. You just multiply atmospheric pressure by the proportion of air that's oxygen. And throughout the planet, regardless of where you are, lower, high elevation, whether you're at the equator or at the poles, all around the planet, the proportion of air that's oxygen is 21% or 0.21. So if you multiply 0.21 by atmospheric pressure, at sea level, you can see here it's 160 millimeters mercury. But at the top of Everest, replace this value here with 250, and it comes out to 53 millimeters mercury. So clearly, the partial pressure at the top of Mount Everest is much lower than at sea level, and you're going to have a very hard time getting oxygen to cross into the body. So let's think about this then with regard to biology. So which of these variables in Fick's law could be influenced by natural selection? So we're trying to get you to think about how natural selection could act to maximize gas exchange. So hit pause, think about this for a minute. Okay, so which of these variables do you think natural selection could act on? So clearly, a variety of animals have different sized areas for gas exchange. So A would be one variable that could be acted on by natural selection to maximize gas exchange. Also, the thickness of the barrier, right? So the thickness of the membrane could be influenced by natural selection to maximize gas exchange. So it can have pressure to decrease, or I suppose, depending on the environment, it could increase, but generally speaking, you're probably going to want to decrease that value. Okay, so in order to maximize diffusion rate, you want a bigger and bigger surface area where possible. You want that membrane or the respiratory surface to be extremely thin. And hopefully you have a large partial pressure gradient, so on either side, right? So you need to have ventilation or countercurrent exchange to really maximize diffusion. So now let's think about this in bacteria and archaea, right? So this is one of the main features that has limited the size of these single-celled organisms. So remember when we talked about not only diffusion, but early in the semester we talked about surface area to volume ratios, right? So in order for respiration to occur, these gases are passively diffusing into and out of the cell. But if the cell gets too big, it becomes very difficult to have efficient gas exchange outside and across those membranes and across the plasma membranes and cell walls. So you can only get so big and have efficient gas exchange. The way you overcome that is, of course, becoming multicellular. So they do engage in gas exchange. It crosses the membrane, it diffuses across, but they remain small. What about in fungi? So remember, now this is sort of a classic representation of fungi, but of course, bear in mind that not all fungi make these mushroom-shaped caps when they reproduce. But just to recall the terminology, so many fungi consist of these sort of thread or hair-like structures called hyphae. They're actually a network of cynocytic cells. So these are cells that frequently lose the membranes in between each of the cells. And so they're lined up next to each other, but they lack a membrane in between. And so they're multinucleate. But in any case, so you have these thread-like, these very small microscopic structures, usually in the soil. They can form mats that are called mycelia. And then when they are reproducing, some of them will then push up these spore-producing structures. And in this case, it looks like a mushroom, but they don't always look like that. In any case, so the hyphae and mycelia that are underneath the ground will engage in gas exchange with pockets of air that are in the soil. So it diffuses across the cell membrane. And then for those structures that are above the ground, of course, they exchange gas with the atmosphere. And again, it just diffuses across. With respect to plants, and what we're looking at here is a cross-section through a leaf. And one thing that maybe should jump out at you is that, now mind you, this is magnified. I don't know at what size, but I'm going to hazard a guess this is probably at 40. In any case, even though the leaf is quite small, you can see that there are several cell layers between one side of the leaf to the other. So that would mean that without any other supplementary structures, gas would have to exchange would have to diffuse through several cells in order to actively get oxygen or to engage in gas exchange in those inner cells. Now, as it happens, plants respire at much lower rates than animals, even when they're actively photosynthesizing. So when they're actively photosynthesizing, they are using quite a lot of gas exchange. But even when they do that, it's still at a much lower rate than animals. Now, what this picture is showing you is that sometimes there are pockets of air spaces within those leaves that will facilitate some gas exchange. But as you know, they have stomata, which are not indicated here, but they have stomata that open and close and allow gas to enter and exit, usually the underside of the leaves, although you can find stomata all over the plants. But as it happens, so stomata facilitate that, but gas exchange can occur across the cell wall at the surface. So these cells that are actually along the edges of the leaves are engaging in gas exchange as well. So to reiterate, so most of the living cells in a plant have at least part of their surface area exposed. And then here I also say the loose packing of parenchyma cells and leaf stems and roots provides a system of air spaces. So that's going to facilitate gas exchange occurring among the adjacent cells to those air spaces. So oxygen and carbon dioxide are going to pass through the cell wall and plasma membrane by cell diffusion. But as it happens, aquaporins can facilitate not just water movement, but the diffusion of carbon dioxide through those same channels. But plants also have some additional methods for engaging in gas exchange or facilitating gas exchange. So if you think about it, what about for parts of plants that are really woody, right? So how do they engage in gas exchange? So in woody stems, they actually have these little things. So what we're showing you in this picture, all these little like sort of rectangular white sort of structures, right? So these are called lenticels. And you can think of them in a way as being like bigger stomata. So these are actually pores through which gas exchange can occur through layers of dead cork cells and waxy waterproof coverings on woody plants. So woody stems and mature roots are sheathed in layers of these dead cells impregnated with suberin. So that's the waxy substance, and it makes it hard to actually, so it helps to retain water, but it hurts gas exchange. So these lenticels perforate the tougher layers, and it enables oxygen to reach those intercellular spaces and to release carbon dioxide. I think I just said that in the opposite direction. So to take up carbon dioxide and release oxygen. Anyway, either way, gas exchange is occurring through those lenticels. Okay, so now let's talk about how terrestrial vertebrates obtain oxygen and release CO2. So in the case of gas exchange in animals, so we're talking about things that are terrestrial. There's 5 steps. So you need to have some sort of air in the environment, right? There needs to be some sort of ventilation. And in this diagram, it doesn't really matter whether it's air or water, but right now we're focusing on terrestrial organisms. So we're focusing on air. So things that are not in the water. So you need ventilation. It's the movement of air, in this case, through a specialized gas exchange organ, such as a lung or a gill. We're going to get to aquatic organisms in a minute. So it needs to diffuse across this respiratory surface. So diffusion is the second step. As the gas diffuses in, it's going to enter the circulatory system. And in this case, we're talking about oxygen entering in, circulating through the body, then diffusing into cells that need that oxygen in order to engage in cellular respiration. So the 5 steps are ventilation, diffusion, circulation, diffusion again into the cells. In this case, it's diffusion into the body and into the circulatory system, diffusion into the cells, and then cellular respiration. So sorry, the bottoms of these slides are going to go through each of that. So diffusion at the respiratory surface, where oxygen moves from the air or water into the blood, and carbon dioxide moves out into the environment. Circulation is the transportation of dissolved oxygen and carbon dioxide throughout the body. Diffusion into the tissues or out of the tissues in the case of CO2. And of course, cellular respiration requires O2 and produces CO2. So if we're talking about vertebrates, most vertebrates have lungs, although if you take my herpetology class, we'll talk about some notable exceptions to that rule. So there are lots of organisms that can respire without lungs. In any case, if we're talking about basic mammals, so air is going to enter your body through the mouth or nose. The trachea is going to carry that inhaled air down into your lungs, into the bronchi, into the bronchioles, and then into the alveoli. So if we zoom in on that last piece, so into the alveoli, so mammalian lungs are divided into tiny sacs called alveoli, which greatly increase the surface area of gas exchange. And so what you're seeing right here is 4. So there's one here, second one, third one, and a fourth one. So these are all interconnected by openings, but you can basically think of it as a complete sort of bubble or balloon. And so in this case, you're seeing 4 of them. So humans have approximately 150 million alveoli per lung, okay? And something else I'd like to point out is not only the flow of air into these pockets, but notice these capillaries that are directly adjacent to these alveoli. So there's only really one cell layer. It's a little bit more complicated than that, but you have the one cell membrane of the alveolus and then the one cell entering into the capillary with a little bit of space between. In fact, let's look at that in a little bit more detail. So if you look at it this way, so here's the alveoli, right? So air is coming in. Now there's usually a film, an aqueous film on top. So you need that aqueous film in order for the most efficient gas diffusion to occur. But here's the one cell layer of the, there we go, of the alveolus. There is a small space, the extracellular matrix. Here's the wall of the capillary, and then you're into the circulatory system. So you have a very short distance that you need to passively diffuse across in order to oxygenate the blood. So how does air enter and exit at least mammalian lungs, right? So we have a really important structure called the diaphragm. And the diaphragm is largely under autonomic control. So it's automatically moving without you thinking about it. Although you actually can control that. Clearly you can stop your breathing should you choose. But when we talk about how air enters and exits the lungs, for those organisms that have a diaphragm, what's happening is what's called negative pressure ventilation. So when the diaphragm contracts, and hopefully you'll recall that with muscles, and we're going to talk more about this later on in the semester, but muscles can only contract. They can't push, okay? So when the diaphragm contracts, it's actually going to move downwards, and that's going to draw in air into your lungs. And then when the diaphragm relaxes, now it doesn't push up, but when it relaxes, it's going to then go back to its original position, which is going to allow air to then exit the lungs. So this is called negative pressure ventilation. So you're drawing air in, and then it's the muscles relax, and then air is forced back out of the lungs. Oh, so this is just the other side of that. So when you exhale, the diaphragm relaxes. It's going to move up, but it's not pushing up. It's just going to its normal. This is actually sort of erroneous. So this is sort of its normal position when it's relaxed. And when it tenses up, it's going to constrict and move down. But when you're exhaling, it relaxes and the pressure is less negative, and then it moves out of the lungs. So ventilation rate, when you're not thinking about it or trying to control your breathing, is under homeostatic control. So remember at the beginning of the semester when we talked about sensors, integrators, and effectors. So this is a system that functions under that umbrella. So when carbon dioxide levels increase, your blood pH is going to decrease. And you probably remember this formula from chemistry. So when this happens and your pH lowers, that's going to be sensed by the carotid arteries and then special neurons in the brain. So there are places that are going to sense that, okay, pH has gone down. When that happens, there's a signal sent to the medulla oblongata. So it's sensed in one part of the brain that pH has gone down, sends a signal to the medulla oblongata that, hey, you know, is this at the right set point? Are we at the set point or higher or lower? The medulla is then going to send a signal to the effectors, right, to the rib muscles and the diaphragm. That's going to increase the rate and the depth of ventilation, and that should result in expelling more CO2 which should then bring the pH up to where it should be for the body. And then, of course, this cycle will repeat. So there's still going to be a detection of, is CO2, sorry, it's actually detecting pH. So is pH, at the set point or not, back to the medulla oblongata, sends a signal to the effector to raise heart rate. Hopefully that releases CO2. And eventually pH should drop back to normal. So if we were in class, we would have a team discussion on diseases that can impact gas exchange. So how do each of these diseases or conditions affect the respiratory system? And so we would have broken it up into emphysema, asthma, pneumonia, and cystic fibrosis. So all of these conditions are a little bit different, but they have some similar impacts. So in the case of emphysema, this is often caused by smoking, but there are some other ways that emphysema can come about. What happens is there's a destruction of your alveoli, and it causes an enlargement in air spaces. So what you can see here is, here's normal alveoli, and they have these sort of like cup-like structures. And remember earlier when we talked about surface area to volume ratio, right? So all of that membrane in each of those cups is surface area for respiration to occur and diffusion of gases to cross. But when you have the destruction of those membranes in between, you end up with one big cup rather than several little cups with extra membranes. So you destroyed some of that surface area for diffusion to occur across. And it causes enlarged spaces, which is actually not good for respiration. So you actually want... So you don't want a single large open space. You want extra membranes in there to increase your surface area for diffusion. I should probably say, often when this happens, there's also a buildup of mucus. And that's no good either, because that's actually going to create a larger barrier for diffusion to have to cross. And so it's going to reduce the rate of diffusion and gas exchange. Similar for asthma, but in the case of asthma, you get a similar impact. But what's happening is when you have an asthmatic attack, there's a narrowing of the airways. So there's like a constriction in the musculature. And there's often mucus that develops as well. So same thing, you're reducing the surface area for gas exchange to occur. You're disrupting the length of or the width of the membrane or the area across which diffusion has to occur. So again, reducing gas exchange in general. Pneumonia is similar. This happens when you have a buildup of fluid in your air sacs. So now it's going to reduce the surface area across which diffusion can occur. Similarly in cystic fibrosis. So it's a different mechanism for why this happens, but you get mucus that develops in the lungs and blocks airways. So just as you saw with pneumonia. So you have a decreasing surface area, decreasing volume of air that's exchanged, and decreasing surface or diffusion across the surfaces. So it's a heavy impact on you. So we talked about mammals. So as it happens, all mammals have a diaphragm that facilitates respiration and getting gas exchange into and out of your body. However, not all animals have a diaphragm, and most non-mammalian animals, in fact, all non-mammalian animals lack a diaphragm. So there are other ways to respire and to engage in gas exchange. And this figure is meant to be highlighting the respiratory systems and muscles that may be involved. But it turns out this is kind of a funky diagram because it looks like we've got unicorns and apparently Godzilla. So kudos to whoever made this diagram. It wasn't me. Okay, so if you don't have a diaphragm, so frogs are a classic model for organisms that respire and have lungs but don't have a diaphragm. So the way that they get air into and out of their lungs then is via what's called positive pressure ventilation. So remember, with negative, you have a muscle that's drawing air into the lungs. In the case of frogs, what's happening is you basically are forcing air into the lungs, okay? And the way that you do that is you can see videos anywhere online where you see frogs. Often it looks like they're kind of pumping their throats, right? So they're kind of, doing this sort of a movement, right? So that's called gular ventilation. So basically their gular sacs, or their throats, are actively pumping up and down. That draws air into the mouth, but then as they, so when they draw their gular patch down, that draws air into their mouth. But when the gular patch goes up, it forces air into the lungs. So what you see here, lowering the floor of the mouth draws air inward through the nostrils, and then closing the nostrils and raising the floor of the mouth pushes air into the lungs. Contracting chest muscles and raising the floor of the mouth forces air out of the lung, and the frog exhales. And so that's why you see them sort of flapping their throats. They're actually actively pumping air into their lungs. So negative ventilation draws air in. Positive pressure ventilation forces air in. Now, birds have a whole different system going on, and so they have a great adaptation for enabling them to get a lot of gas exchange going on in order to power their high metabolism. So birds, generally speaking, have incredibly high metabolism, and they're incredibly active. So all that flight, it costs an extraordinary amount of energy. And in order to be able to do that, they need to be able to get oxygen into their bodies and quickly. So they have a really efficient system that allows oxygen to constantly be flowing across their lungs, allowing gas exchange to occur. So it's referred to as being unidirectional. And let me show you how this happens. But what's amazing about the system is that they're able to engage in gas exchange during inhalation and exhalation, unlike what we do. We inhale, gas gets, diffuses across the membranes. We exhale, all that gas goes away. So there's no more diffusion going across, right? So it's only when we inhale. These guys, it happens when they inhale and exhale. So this is a stylized diagram of a bird. I guess it's meant to be a duck. And so you can see the entrance into the trachea, but look at all this. So if we zoom in on this, so here's the trachea. Here's what's going to happen. So it's going to be a sort of 2-cycle process. So they're going to inhale, air is going to immediately go into these posterior air sacs. There's really not much for gas exchange happening in those. When they exhale, air is going to flow from these positive air sacs, these positive posterior sacs. Sorry, I can't get it out. From the posterior sacs into the lung. So these layers are called parabronchi, and this is where all the gas exchange is occurring. So diffusion is happening across this highly vascularized set of sacs. Then, when they inhale for a second time, gas then moves out of the parabronchi and into these anterior air sacs. And then when they exhale for the second time, air goes out. But what's happening is, remember, each inhale, with an inhale, the air that's going into these positive air sacs, there was already air there. So that's already going across the parabronchi. Then they exhale the first time, air from here goes in the parabronchi. Inhale the second time, air moves back here. Air that was in the parabronchi move into the anterior air sacs. And then when they exhale the second time, this air goes back. You get the picture, right? So there's constantly airflow going over these parabronchi and across the lung to engage in gas exchange. So they inhale and exhale. There's constant gas exchange happening, and it's a hugely efficient system to power their energy needs. Okay, so I said we were going to talk about aquatic organisms too. We've talked a little bit about this, but let's go more into what's happening with fish and exchange across their gills. But remember, fish aren't the only ones with gills. There's other organisms as well. So gas exchange in water is made a bit more difficult because there's a lower amount of oxygen, available oxygen in water. So there's less air in water than in air. There's less oxygen in water than in air. And water as a media is pretty dense. So it's a little harder to move oxygen through water than it is through air. But gills are specifically are supremely designed to be able to do a gas exchange in an aqueous environment. But in order to do it, you're going to need a huge amount of surface area, and you're going to have a well ventilated system. So you're probably aware that with many organisms, if they're not actively moving water across their gills, if they're just sitting in place, they could in fact drown, as it were, or basically use up all the oxygen in an area and then no longer be able to respire. So they constantly have to move water across those gills, either by actively moving and swimming, or by opening and closing their mouths and letting water flow over their gill surfaces. So there's two kinds of gills, in case you didn't know. So there are some organisms that have external gills, and so you're looking at things like tube worms. There's all kinds of worms that have, aquatic worms that have external gill structures. There's amphibians that actually have external gills. And then there's a whole host of organisms that have internal gills. And by that, we really just mean that they're covered up. They're not necessarily on the inside of their body, because if you lift up their gill covers, you can see them. This picture happens to sort of highlight what internal gills are like, but it's a little disturbing because what's happening is they've removed the carapace on the side of this crawfish so that you can see the internal gills. And this is kind of what you find with fish too. You can sort of pry up the opercular cover and see the gills. Wow, there's music with this video. I didn't realize. Anyway, fish gills are located on either side of the head. And you can see here in this video, this fish is actively opening and closing its mouth. And it's a little harder to see, but it's actually moving. There you go. You can see it moving the opercular cover right here to allow water to enter the mouth, flow over the gills, and then exit out the backside of the gills. So when they're doing this, they're creating a pressure gradient that moves water across those gills. Okay, that music was really bizarre. Anyway, let's look at this in a little bit more detail. So this figure is showing water moving in the mouth, across the gills, and then it's going to exit out the backside of the opening from the gills. So what you have here are a series of structures, right? These are called gill arches, sometimes called gill rakers. And so these cartilaginous structures are actually providing a surface where gills are attached to them. They stay in contact with the water. They must be moist in order for respiration to occur. But they must be allowed to open up and not collapse on each other in order to, if they collapse on each other, they're going to reduce the surface area that allows for diffusion to cross. So they're going to extend out and into the water column. So here's those same structures close up. Here's those gill filaments. You can see these finger-like structures, so these gill filaments. And if we zoom in on a gill filament, here's what it looks like, right? So there are these rows of what are called lamellae all along each of the gill filaments, and here they are. And what these are then is it's a very thin membrane that's highly vascularized. And so you can see these capillaries that are within the lamella. And so there's one cell layer between them and the respiratory surface. So water is flowing across. Oxygen is being withdrawn from the water into the capillaries, and it's then going to be transported by the arteries back into the body. So here's oxygen poor blood that's coming in, filling into these capillaries. The blood is moving across the capillaries, picking up oxygen, and then it's going to be transported through the vein back to the body. Let's take a little bit closer look at what's happening with partial pressures here. Let's draw this together with that partial pressure discussion. Okay, so here's what's happening. So here's the gill lamellae. And notice, traditionally, usually we indicate blue when it's oxygen poor and red when it's oxygen rich, okay? So in the case of gill lamellae, what's happening here is what's called countercurrent exchange. So you've got water that's flowing across the gills here, right? And notice that, so as water is coming, so water is going to be oxygen rich, And then as it exits the gills, it's going to be oxygen poor, right? Because oxygen is going to be withdrawn out of it and into the gill lamellae. Okay. Oxygen poor blood is coming from the heart into these lamellae. Here's the capillary bed. So it flows through this artery, across the lamellae, and then out the vein. Okay. Now, sorry, I should probably back up and say, so what's going to happen then is that as water approaches the gill lamellae, there's going to be this incredible diffusion, high diffusion of oxygen across the gill lamellae, oxygenating the blood. And then as the water continues to flow, it's going to have less and less oxygen. And so even though the lamellae will engage in diffusion, as the water flows across, there's going to be less and less oxygen, and so lower and lower diffusion rate. And there's another way to actually show that to you. If you look here on this left-hand side, this is countercurrent exchange. So this is water flow over the lamellae, and it's showing percent oxygen. So when it first reaches the gill lamellae, there's 100% oxygen, right? And then as it's flowing across, that oxygen percent is going to decrease naturally. This is blood flow through the lamellae. So right when it first crosses, there's 90% exchange of oxygen, right? 60%, 30%, 5%. So as you're looking at flow through the lamellae, it gets to be less and less and less. And that's a natural response of oxygen diffusing over the lamellae. So this is just a figurative way of depicting what's happening right here. Okay? Now, we're not going to talk about concurrent flow. We're going to come back to this in a later lecture, so don't worry about this part for now. So fish gill is a countercurrent system. So the advantage is that there's a slight gradient in partial pressure of oxygen between the water and the blood, and it exists along the entire length of the lamella. A large difference in oxygen partial pressure exists between the start and the end of the system. So what's happening then is that most of the available oxygen is absorbed as you get across. So it's basically sucking all that oxygen out of the water. And that's a really efficient system. We're focusing mostly on the advantages here, but you can imagine one of the disadvantages is, again, having to deal with that low oxygen rate compared to air, and the density of the of water versus air. But fish gills eloquently overcome this these disadvantages. The other thing is they need to keep that water flowing, so they can't stop pumping water over their gills, and or they can't stop swimming, at least for very long periods of time. Okay, so how does gas exchange occur in insects? Well, we started to talk about that last period. So we talked about a tracheal system of respiration and these openings to the outside, these spiracles that they can open and close to allow to allow gas exchange to occur. Here's another example. So last time we looked at a grasshopper, here's a bee. And then in blue, we're showing you the tracheal system and then where the spiracles occur. Sometimes they're not very easy to see if you're looking at the organism itself unless you're looking under a microscope. So we've kind of covered this already. So the air-filled tubes are called tracheae. They open to the outside through pores called spiracles. Air moves from the atmosphere through the spiracles into the trachea. And again, there's no active pump to do this. There's not a diaphragm. Basically, just by virtue of the muscles in their body contracting as they move. So when they contract, it sort of increases pressure in the body that forces air out. And then when they relax, that allows air to come back in. So the diffusion of oxygen from the trachea into the cells is not associated with the blood supply. By the way, you should probably recall that these guys also have an open circulatory system. So there's hemolymph that's going to be surrounding that tracheal system. So in larger species, the volume of the tracheal system changes with muscle contractions and relaxations, which produces pressure changes, which is what I was just saying. And so what you're seeing here is these tracheae are dilated here, allowing air in, and then they get compressed here due to muscle tension. Now, so I mentioned last time how the tracheal system allows for gas exchange, but it's a somewhat passive gas exchange. And as a result, it's going to limit how big insects can get because in order to efficiently respire, they need to be able to get oxygen in and out. And that tracheal system makes it difficult given the amount of oxygen that's in the air right now. But in the Paleozoic, oxygen concentrations were far higher than what they are now. And as a result, that means you could get more oxygen diffusing across than you would have in the past. So what could occur? That's right, giant insects. And so this is a fun little graph from the web. Presumably, I don't know who Ripley is, but apparently it's some kind of soldier dude. Anyway, with higher amounts of oxygen in the atmosphere, you could get really big insects like this largest known Meganeuropsis permiana. And actually, you can get some pretty big insects, like stick insects are pretty big, even modern ones today. Here's how big the biggest of our current dragonflies are. So you can see it's far smaller than this guy here. And even some other insects, if you look by comparison, the Titan beetle, the Goliath beetle, and the giant Weta, they're pretty small compared to some insects that were really big. So that's kind of a fun fact to leave you with. Okay, that's what I'm going to leave you with. We'll talk more. I'll send you some emails regarding the study guide and things coming up next week. And if you have any questions, send them to me. I'll also send you a survey regarding when we can do a review session. And get studying. We got a test coming up on Tuesday. Stay warm.